Tyndall. I'm the Manager of Digital Innovation here at the University of Adelaide Library. I'm also one of the co-conveners of the coordinating group for the Artificial Intelligence for Libraries, Archives and Museums Australia and New Zealand chapter. It gives me great pleasure to welcome you here today. Before we get started, I'd like to acknowledge that I live and work on the lands of the Kaurna people. And I want to acknowledge and pay my respects to them as the traditional custodians whose ancestral lands we virtually gather on today. I acknowledge the deep feelings of attachment and relationship of the Kaurna people and country, and respect and value their past, present and ongoing connections to the land and cultural beliefs. We have people joining us today from across Australia, Canberra, Hobart and others, just to mention those that are here on the panel, and the world. And I'd like to extend that acknowledgement to relevant communities who are represented here today. And with that, I'd just like to hand over to my friend and colleague, James Robertson from Digital New Zealand, who will welcome us on behalf of our New Zealand friends.
Thanks, Alexis. Kia koutou, ngā maonga, ngā awa, ngā waka, ngā tupuna o Aotearoa. Me te whenua moe moe e hui hui mai nei, tēnā koutou katoa. To you, the mountains, rivers, waka, ancestors of Aotearoa and the land of the dreaming that are gathered here, greetings to you all. Thank you. I'm Systems Manager at Digital New Zealand, the National Library of New Zealand, and I'm very pleased to be here and helping, especially Alexis and Ingrid, manage this presentation. Thank you.
Thanks very much, James. So welcome to today's webinar, Computer Vision and Automation for CSIRO National Research Collections, the latest in our series of webinars from the Australian and New Zealand chapter of AI4LAM, or Artificial Intelligence for Libraries, Archives and Museums. Welcome back to our repeat attendees. I saw quite a few familiar names on the list, and a very special hello to anyone who's joining us for the first time. We actually had a massive response for this session, smashing our attendance numbers from previous webinars, with more than 150 people signing up, which is wonderful to see. If you are new to this community and you're interested in further engagement with AI4LAM, at the end of the session, we'll share some links and some information about how to get further involved. In terms of housekeeping, I'd just like to make sure you know that today's session will be recorded and shared online after the event. And I'd like to invite you to introduce yourself in the chat window. I can see some people are already doing so. And if you are interested in sharing any comments about your interest in these issues, please feel free to do that too. That will be the place for asking questions during and after the presentations. And we've got Ingrid Mason online who'll be keeping an eye on that area and making sure your questions are addressed. Housekeeping done. We've got a great set of presentations for you today. We're really pleased to have collected with the lovely folk from the CSIRO National Research Collections Australia. This extraordinary group care for our national insect, fish, wildlife collections, our algae culture collections, national herbarium and the Australian Tree Seed Centre. They look after an absolutely invaluable record of Australia's environment and its scientific record. And it's very exciting to see them connecting with CSIRO's machine learning and artificial intelligence future science platforms initiative to look at new ways of looking after and using those collections. I won't go too deeply into it because we've got a fabulous range of speakers for you today, and I'm sure they'll tell you all about it. I just want to introduce all three of our speakers today before we start, and then we'll flow through the webinar as we go. So first up, we'll have Dr. Pete Thrall, who's the Group Leader for Digital Data and Informatics at National Research Collections Australia and Chief Research Scientist for CSIRO National Collections and Marine Infrastructure. Over more than 25 years, Pete's research is focused on the ecological and co-evolutionary dynamics of natural host microbe interactions using theoretical and spatially explicit computer modelling, experimental and field studies and molecular approaches. He's published and presented on these scientific interests in a range of academic and popular environments and is now applying that wealth of experience to these challenges that face the National Research Collections. We'll have Dr. Chris Jackett, a postdoctoral research fellow who's working in object detection activity and microscopic imagery. His PhD research invested a range of mathematical, statistical and computational mechanisms for improving the quality of satellite data. And he complemented that study with a graduate diploma in marine science. So the perfect combination for these things we're talking about today. He'll tell you about the work that he's doing on developing machine learning algorithms for automated detection of harmful algal bloom species in microscopic imagery. And we've also got a presentation from Dr. Abdul Wahid Khamis, who's a CSIRO Early Research Career Postdoctoral Fellow. He's working on developing machine learning algorithms for visual object detection in natural collections, including plants and insects. His doctoral research focused on the use of RF technologies for medical sensing applications and resulted in a number of innovative contact-free sensing systems for hand hygiene tracking and vital sign monitoring. So with that introduction, and they are joined as well by Nicole Fisher, who's one of the collection managers for National Research Collections Australia, who's also going to be available at the end for to make sure that we've got a proper answer to all any questions that you might have. With that, I'm going to hand over to Pete, and he will get us started. Thank you.
Great. Thanks, Alexis. Just checking to make sure you can hear me.
Yep.
Yep. Okay, good. Let me just share my screen, see if I can get it right. Okay. So just check with you, Alexis, whether you can see the presentation.
Yes, that's perfect. Thanks.
You can. Okay, great. Thank you. So thanks for that introduction, and thanks everybody for having us along. We're looking forward to talking to you about some of the work that we're doing here at CSIRO, focused on biological collections. And so I'm going to start us off by giving a little bit of just a general overview of what the collections at CSIRO are all about, some of the challenges, I guess, that we face, and a little bit about some of the directions we're heading in terms of digitization and utilizing MLAI approaches. And then Chris and Abdo will give you some more specific talks on some of the projects that we're doing using such approaches to actually get more out of the data that are embedded in these collections. So let me just go to the next slide. Okay, so Alexis mentioned the biology collection. CSIRO actually manages a whole range of different kinds of collections, ranging from the ice cores that have been collected to as part of the climate science that we do, to the National Soil Archive, air samples, foodborne pathogens, and of course the biology collections that we work on. So all in all, CSIRO manages probably some 50-odd different kinds of collections across the organization. So it's part of our role, I guess, as National Research Agency to maintain and utilize and secure that piece of national research infrastructure that's represented by the collections. But what we're going to focus on today is specifically the biology collections, which all in all represent somewhere around 15 million or so specimens. We have a number of significant collections. There's a wildlife collection, the fish collection in Hobart, where Chris actually works, the insect collection, the National Herbarium, Tropical Herbarium up in Cairns, and then the Tree Seed Center and the algae collections, which are both a little bit different from the others in that those are primarily living collections rather than dried or preserved specimens. Those collections, there also is an enormous diversity of types of material in these collections. As I mentioned, some of them are living collections. There are plant specimens, specimens preserved in alcohol, insects mounted on pins, X-ray, photographs of fish. In some cases, we have actually extensive slide collections, microscopic slide collections, several 100,000 of those. Genetic data, sound libraries, for example, for bird calls. So there's just an enormous diversity of different types of data that are embedded in these collections. And it's a huge challenge, actually, just managing all of that different types of material, not just managing it, but then sort of actually utilizing it, because of course CSIRO is primarily a research organization. So these collections are meant to be used for research purposes, etc. Just, I guess, to set the scene a little bit more, just recently we went through a process of developing a new 5-year strategy for the biological collections, which is really built around on 4 main pillars, I guess you could say. Securing the collections, which is about managing and maintaining those collections. And in fact, we're in the process of building a new facility here in Canberra to house a number of the collections together, to bring them together, to enhance the value add to those collections, to better enable how we utilize those collections, to mobilize them, to deliver more, and to address national and international challenges. And of course, increasingly, and for those of you online who also work in biology collections, increasingly biological collections are being asked to contribute and to help with a range of challenges around biodiversity discovery, future industries, biosecurity, conservation, planning, and so on. So there's just a whole range of areas in which collections are increasingly being asked to sort of play a role in addressing these various challenges. And digitization and new analytic approaches, for example, machine learning and artificial intelligence, play a role across all of those areas. And so the group that I manage is actively working across the biological collections managed within CSIRO to tackle all of these kinds of things. So we're looking at the way that we digitize or image material. We're developing and moving into new systems to better integrate how we handle our data, and then looking at new analytical approaches and tools that are based on machine learning and artificial intelligence. And Chris and Abdo's talks will both really focus heavily on some of those analytical approaches that we're utilizing. We're currently engaged in many different kinds of digitization activities. One of the projects that we're working on is to catalog all of the type specimens, to digitize all of those, in part to ensure the security of those really valuable specimens, but also to make them more accessible. Sound libraries, I mentioned already, we've got extensive sound libraries for many of the bird species in Australia. One of the projects that we're currently actively engaged in, and this is only likely to grow into the future, is the utilization of MLAI approaches for biosecurity applications. And in the upper right-hand corner of the slide is just a picture showing one of those projects, which is focused on a particular group of pests, stink bugs, brown marmorated stink bugs, in fact. And basically, some of the researchers within the collections have developed an app which can be used to identify particular pest species in this group using machine learning models to do that. And in fact, we're looking now to actually expand that sort of work. It's something that's getting a lot of interest externally from the department, and clearly is something that would have potential in terms of deployment, for example, at borders and so on. And then we're currently also doing some work with the department, again, the Department of Agriculture, Water, and the Environment, to look at post-fire impacts on rare and threatened species of invertebrates, and doing some imaging work there. And that's in the context of making that data available for citizen science inquiries. One of the projects that I'll mention in particular is a project we're currently engaged on, and this is something that I know is occurring at many museums and biological collections worldwide, and that is looking to use mass digitization to really start to tackle the enormous, I guess, backlog of material that currently sits in collections. And so we've actually engaged an external provider who specializes in herbarium mass digitization, Picture, based in the Netherlands, to fully digitize and image the entire herbarium collection that CSIRO manages. That's 1.2 million specimens in total. And we're just now in the process of preparing for that, with a likely kickoff of that project in January. But basically, by the end of next year, we expect to have fully digitized the herbarium collection, which means that collection then becomes accessible and discoverable and usable, not just within CSIRO, but to people externally as well. For example, through the Atlas of Living Australia, which is a data aggregator that we can use. Of course, digitizing all of this material also brings enormous challenges in terms of managing it. How do we use it? How do we make better use of it? And so on. And MLAI approaches start to come into play, I think, when we start to talk about some of those challenges. So we know we can't do everything. And one of the issues that we have, and I'm sure it's common to everybody online, is the collection staff and taxonomic expertise, or the equivalent in other types of collections, are in short supply. And so that means that there's a real backlog in terms of species identification, in terms of getting stuff databased, in terms of tackling the analytical science around how we utilize those specimens. What do we prioritize is a question that think we constantly wrestle with, and probably others will as well. We're also working to better integrate the collections data, the huge amount of information embedded in collections, with the broader CSIRO data ecosystem. So that gets into issues around data management, storage solutions that we're looking at, and then how we make that information discoverable and usable for researchers and end users. And finally, as I mentioned, there's a number of global biodiversity initiatives, the Global Biodiversity Information Facility. There's initiatives in Europe and North America that are focused on digitizing biological collections. And so we're sort of working to link what we do with those broader initiatives as well. And it's all about really keeping biological collections relevant for the future, so that we don't want these collections to just be seen as the sort of traditional sort of collections that are sort of more static kinds of facilities, but really that are very active elements of research, active elements of addressing some of the environmental problems that I mentioned earlier, and going forward. So that's one of the sort of broader goals that we have. So I just wanted to mention a couple of things that we're doing. One of the things that we're doing is looking at how to better integrate data management, and that has a couple of, I guess, threads to that. One is simply just how do we better manage the data itself, but also there's a growing interest in the biological community in what sometimes is referred to as the extended specimen concept, which is really about how do we link multiple associated data layers that might be relevant to a specimen, so genetic layers, phenotypic data, environmental data, geospatial data, etc. So that really building up a much more complete set of information around a given specimen, and then being able to utilize that information for research. The second thing I wanted to mention is actually quite new. It's I think got a lot of potential for collection curation and collection science, and that is we're just in the beginning stages of developing a project to look at the potential to integrate digital and human intelligence to assist with collection management. And the sort of idea is that machine intelligence is really good at some kinds of things. Human intelligence is really good at other kinds of things. Can we develop a system or an approach where we're actually getting machine and human intelligence to work collaboratively and interactively to assist us with collection management, which might help us to address some of those issues around the shortfall in human curatorial capability. And it could be really for a range of things, helping us with actual managing databases in terms of data quality, being able to look for anomalies, almost having a digital curator that could look over database entry sort of people doing database entry, looking over their shoulders a bit and ensuring that we catch mistakes early. We're looking at the potential to utilize digital curation to assist with early initial species identification and classification. There's a range of ways in which we sort of think that a digital curator could become a really invaluable, I guess, member of a collection team. So we're just about to kick off a project looking at how we bring human and machine intelligence together to do that. Really at the very early stages of this, so I'd say stay tuned to see where it gets us to, but we're hoping over the next few years to be able to trial the concept of a digital curator across the CSIRO biology collections. Okay, so that gets me to my last slide, actually, which is really just, as I said, a bit of a general introduction to the other two talks. And what I want to do now is basically hand things over first to Abdo, who's going to talk, whose talk will be about trait extraction from insect and plant specimens. So the goal of using this, for example, to aid with identification, which could potentially be integrated into this broader concept of a digital curator. And then Chris Jackett, who will talk to us about the use of machine learning and AI approaches in the detection of harmful algae. So I think, Alexis, with that, I will stop sharing my screen and hand it back to you.
Yeah, thanks, Pete. I will. Abdo unfortunately wasn't able to be with us live today, but we have his presentation, which we will share for you now. Let's just check. There we go.
Hello everyone, this is Abdo, and today I'm going to talk about extracting traits from digitized collections. I was hoping that I can do this presentation live, but this might not happen, and if this is the case, feel free to reach me via email or my LinkedIn profile in case you have any questions about the presentation content. You can find the QR code for my LinkedIn profile here. Just scan it using your phone if you have the LinkedIn app. In today's talk, I'm going to go briefly over the problems I'm considering as a part of the object detection activity in CSIRO. And my focus is on the herbarium and the insect image. I'm working with experts from the two domains, and together we are aiming to find an automated way to do the trait extraction using the help of machine learning. I will explain. Let me tell you first about the problem. So we have digital image that can contain some data about the insects, or for example, it can be a herbarium sheet. And from these two, we are interested in extracting some specific traits. The list is very big, but we are interested in something like the color of the body part of the insect and the area and the size, and also something similar for the plants. So we are also interested in something like the leaf length and width, the area, the color, and how it is shaped, and so on. The way this is usually done is that the experts spend many hours doing this kind of work manually. So they go over the digital samples and do some kind of manual estimation of all these traits to allow them to do further analysis based on these data. And our hope is that we can involve machine learning and AI techniques to take some or most of the manual effort spent here. And of course, the big question that I'm concerned with is what kind of problems that we will attack, and what kind of techniques that we'll use from machine learning to do this kind of stuff. For today's talk, I'm going to focus on one of the things that I find very interesting for me, and it is about shape information. So there are many ways we can take a digital input like this and extract information from it using machine learning techniques, provided that we have the proper amount of data and the proper annotations and labels. And for example, in these two images, you might have a machine learning system trained to predict the location of the leaves in the herbarium sheet, for example, or the head of the insect. And the usual way that this is done is that we train the model using lots of data, and we provide annotation. So for example, in this sheet, we might give this sheet as an input, but we have some kind of a mask over each single leaf, telling an object detection module or model to focus only in the parts where the leaves are, because this is the thing that I'm interested in extracting. This is something very mature now, but it has its own cost, because you will need to spend lots of time doing the annotation, and you will need also a large amount of data. But what I was thinking of, rather than going this traditional direction, we can at least modify it or complement it with additional information. So here, and this is an illustration of how we do the process using the fully supervised approach, in which we provide many examples. I call it here learning from examples. So you give an example, many examples to the machine learning, the neural network model, and you train it based on this data, and during the test time, you expect it to be able to recognize the same pattern. When I was working with the expert, I found that they have some kind of proper terminology to describe shapes. So this here is an example from PlantNet dataset maintained by the New South Wales, and they have many herbarium sheets, and they have also some kind of metadata that you can see here on the description. And on the bottom, I'm just highlighting some of the terms that attracted my attention, and turned out later that these terms have some specific visual meaning. So these are some of the examples. For example, when I say chordate, it means that I have a leaf that looks like heart. And you can find a very big list of these shape descriptors, as I call them, for reasons that will become obvious later. You will find a big list of them, and the question that I had on my mind when I saw this: can we get the neural network to understand what is actually chordate is, or alternate is, or ovate, and not necessarily relying only on this kind of information, but it can be additional piece of information. So assume that we go back to the original model that I just showed 2 slides earlier. And then instead of feeding it with an example, you might be able to feed it with descriptions like this. And one way that you can do this, if the model is not able to learn directly from just description, because it's a very challenging problem, you might be do something like shape generation, which means that if you have some kind of model that's able to convert this description into an image by synthesizing it, you can have this kind of output that can be used with any traditional networks that's trained to learn from examples. So this shape generator can be just a proxy. So this is something I'm thinking of creating, because once you have this synthetic data, you remember you will have the labels for free, because you don't need to do the manual segmentation here, because you already synthesize the data yourself, so you can generate the labels as well. And in this way, you can train the neural network on large amount of data, provided that you have the capability to generate shapes. I will touch on this specific point of synthesizing dataset at the second part of my talk, but the key thing that I am going to focus on here is the shape information. So 2 questions I'm interested in. Number 1 is measuring the distance between shapes. And by measuring the distance, I mean if I have, say, 2 leaves or 2 shape of insect wings, I want to know if they belong to the same class or not, based on the assumption that if the shape is very similar, then they are very likely to be in the same class. And for shape encoding, I'm interested in converting the shape to some kind of description. In this case, it will be textual description called an L system. I will explain more about this. And this is the kind of information that I can use for synthesizing data that I talked about earlier. So let me focus on the first point. So distance between shape, the key problem that I'm considering here is illustrated by this figure. So I have here some pre-processed data of insect wings, and as you can see, those are different wings, so the shapes have very obvious differences. And my question is can I measure the distance between them in some, can I quantify the distance between them in some way? In the literature, you will find many ways to work across this problem. So people usually do some kind of feature extraction. So you can go over the details of these images, do some kind of analysis. For example, here, you might consider that this shape is some kind of network, and do some kind of network analysis, which will require you to find all the points of intersection and do some kind of measurement usually used in the network analysis, or do some kind of contour analysis, which you're going over the outside contour and estimate the curvature, which will give you some metrics that might be useful. There are many other features, but the way I see these things is that instead of directly comparing the shapes, they are comparing the features. So they extract some kind of feature from each sample, and if the features are close to each other, they mean that they belong to the same class or not, depending on the kind of features that you're extracting. And instead of doing that, we are thinking of solving the problem directly by measuring the distance between the actual shapes themselves rather than the feature. This will require more illustration. So I will give you a simple example to explain what I mean by this. So here we have the two shapes, and assume that rather than having the insect wing here, we are having a set of point clouds. Those sets of point clouds are organized in some way. So the red ones represent shape 1, and the gray represent shape 2. And you're asking the question, what's the distance between the shape? If you have a way to align them together, and then you measure the distance between the components that are aligned with each other. So for example, each point will be matched to another point, and you have a way to measure the distance between these 2 points, then the distance can be considered the cost of converting the first set into the second set by summing over all the distances. And to explain it in a simple term, I want to convert the first point set to the second point set, and I am asking the question is, what would be the minimum cost to do this? And I consider this cost to be the distance, to be distance between the actual shapes. The key thing also about what I'm saying here is that we can also know the correspondence. So for example, not only I can measure the distance between shapes, but I can also know that this particular point corresponds to this particular one, which means that you can go to the details of the shapes and you can know which part of the shape belongs to which part. So you are actually kind of moving one of them literally to the other, and this can be useful in many applications. But for here, I'm going to focus on the case of classification of the wings. So what I just explained actually can be achieved using some method called optimal transport. Unfortunately, I won't be able to get into the details, but it's basically solving an optimization problem over the mapping and the distance. So it finds what kind of optimal mapping between each of the points that I just showed to you to another point in the other shape. And the following summation gives you the actual cost, which will be our distance in this case. So, but how do we go by converting the shape to a point? Here is how I do it. So in this case, I have some kind of pre-processing to find the center point of each cell. It's a simple pre-processing. It doesn't require heavy machine learning algorithms, so just image processing is sufficient. By locating the center point of each component, I can take the component and convert it to a point, but I also make the size or the radius of the point proportional to the area, just to make sure that those here are bigger than those here. So you will find this set of points normally bigger than those, okay? And then I do some kind of mapping. So here is where I use the optimal transport to do the matchings that I mentioned before, from this one to this one. So those are samples from 2 images, and the algorithm iterates by moving the points gradually. And one interesting property about it is that it doesn't require you to have the same number of points. So as you can see here, the number of points here is much more than this. However, it can align perfectly at the end, and at the end it will give you the correspondence as well. We tried this on some of the public dataset. Here is an example where each cell is calculated by its area. So the darker the bigger the area. We also converted to the same points that I showed you previously here, and do the matching. And this gives you some kind of the optimal transport map, where it says that this particular cell in the first wing will be mapped to those points. So if you see a darker color here, it means that this point is expressed between multiple points over the target cell. So this is the source wing, and this is the target wing, which means shape 1 and shape 2 in our context. This might not be very intuitive, but you can understand it from this illustration here. So after the matching, these are 2 examples. We did the matching, and it tells you that this particular cell will be mapped to the corresponding one in the bottom. As you can see, this is the point of correspondence that I mentioned, where you can have information about which part belongs to which part, and doesn't necessarily mean that you need to match one to do one-to-one matching. Okay. And the actual distance is very encouraging for our purposes for classification. This is preliminary results, but we intend to investigate more in this. So for the same class, the distance will always be smaller than different classes, which means it can be used as an easy way to do the classification. So this is for the first part. The second part, I'm going to talk quickly about the shape information, the shape encoding, and to give you an idea of what I mean by this, consider the following two examples where you might find it very difficult to put into some kind of if you want to describe it to someone, you will find it very difficult to give him a description that satisfies the visual image that you see here exactly. But one way that people consider the literature is doing some kind of an expression or a descriptor. One of the most popular ones is called Fourier Descriptor, which means that basically you describe the contour of the outer contour of each of those shapes using this equation. It has a nice geometric interpretation. I illustrated here by this example. It shows you that you can have this is just an illustration of the Fourier descriptor using some kind of sequence of circles where the speed of the arrow rotation is proportional to the frequency itself. What I'm hoping for is going one step beyond that, because rather than describing the outer contour, I'm interested in the whole details rather than just the outer shape. And what I'm going to consider here is then something called the L system. It has been used in biology and has long history. And the key thing is that if you have something in mind, you can describe it using the following axioms. These symbols, F plus, minus, and the branching can be used to construct various kinds of shapes. So just one example I'm going to show you here. So the expression on the top here can be translated to this figure. So, and those are different expressions that will give you the image that you see to the right of the box. It's very flexible, but usually people go from the expression to the image. So they have an expression in mind and they render it to an image for purposes of studying or modeling and so on. What I'm hoping for is going in the other direction. So this is something that we are currently investigating, and our goal is to come up with a model that generates more expressions. This is a research question that's taking most of my mental power. But let me just show you an example of how this can be useful for synthesizing data, the point that I mentioned in the early of my presentation. So these are different expressions, and as you can see, they are very concise, that they can fit in a single box on the bottom. But once you render them, this is some kind of software for rendering L system expressions. You can have something very detailed and rich, like those videos, and this in fact can be useful for generating some kind of synthetic data that can be used for training. So thank you for listening to my talk, and as I mentioned before, if you have any question, feel free to reach to me. Thank you.
All right, and with that, I will invite Chris to take the stage. Thank you.
Thanks, Alexis, and hello, everyone. I'll just start a screen share and hopefully pick the right screen. There, how's that? Is that coming through, Alexis?
Yep, I can see the right screen.
Okay, well, greetings everyone. My name's Chris, and like Abdo, I'm a postdoctoral research fellow at CSIRO in the Machine Learning and Artificial Intelligence Future Science platform. I'll just move that window out of the way. Today I'm going to talk about automated detection of harmful algae and toxic blooms. So I thought I'd start with a short video that we prepared for Mars 2021, which is a CSIRO organised machine learning and artificial intelligence conference. All of the postdocs were asked to put together a short video package targeted at a 13-year-old science-interested student, and this is what we submitted.
Algae is everywhere. Just about anywhere in the environment there is water, there is also algae. From microscopic forms so small you might not even know they're there, to giant kelps in coastal marine forests. Algae have been found in soil, snow, hot springs, on animals, corals, jellyfish, and even in clouds. Algae are really important to the environment and can come in many forms, including free-floating individual cells, chains of cells, and amorphous blobs. They can even form large structures such as seaweed. In the presence of sunlight, photosynthetic algae produce most of the Earth's oxygen supply. It's interesting to think that every second breath you take was entirely produced by photosynthetic marine algae. Here at the Australian National Algae Culture Collection in Hobart, CSIRO scientists undertake world-leading research into algae. In our living collection, we maintain over 1,000 strains of microalgae. Right now, our scientists are investigating how algae can produce healthy omega-3 oils, improve food crops, support the aquaculture industry, and even create cleaner biofuels for the future. But some types of algae are dangerous and can grow out of control, forming harmful blooms that can damage aquatic life. These types of harmful algae produce toxins that are poisonous to other organisms. Filter feeders such as shellfish continually filter the surrounding water and can end up containing high levels of toxins. Eating this contaminated seafood can make you extremely sick. Being able to identify harmful algae is really important. At the moment, water samples are collected in the field and transported to city-based laboratories. It then takes well-trained, expert scientists looking down a microscope to figure out what type of algae is in each sample. This process takes a long time and mistakes can easily be made. We're investigating how artificial intelligence and machine learning can be used to teach computers how to automatically detect different types of algae. And the stakes are high. Here in Australia, cyanobacteria, which are closely related to algae, were involved in vast numbers of fish deaths in the Murray-Darling Basin in 2018 and 2019. On the east coast of Tasmania in 2012, a large harmful algal bloom forced the closure of the seafood industry, costing Tasmanian businesses millions of dollars. To better manage these events, we're now taking millions of photos of our living algae collection and training computers to recognise important types of harmful algae. This will help us figure out what types of algae are in the environment while on location, rather than having to collect and move water samples and have trained scientists identify algae in the lab. Developing these machine learning techniques will allow us to detect harmful algae much faster, improve the management of the seafood industry, and keep people safe. So next time you're out in nature, spare a thought for the millions of microscopic algae that are essential to the planet and are keeping us all breathing.
All right, now let's see if I can get out of video mode. Yes, so as mentioned in the video, this work is being undertaken in collaboration with the Australian National Algae Culture Collection based in Hobart, who maintain a marine and freshwater algae collection of more than 300 species and 1,000 strains, so multiple strains per species. And geographically, the collection comes from as north as tropical Australia to as south as Antarctica, with the Antarctic strains being held in a slightly cooler refrigerator. But we're really interested in leveraging this immensely important biological collection and imaging it and developing machine learning techniques for the rapid detection and classification of harmful algal species in marine environments. So the reason this is important is because in the presence of concentrated nutrient sources, algae will grow exponentially and form harmful algal blooms. These large-scale events do happen, and they can cause a pretty serious and damaging impact on the environment and aquatic organisms. So some species of algae produce toxins that are poisonous if you come into contact with them, while other species will strip the water column of oxygen. So if you're an aquatic organism and you move across the front of an algal bloom, you will find it pretty difficult to breathe very quickly. So at the bottom, we've got a couple of images of harmful algal blooms, and they've been described a bit like an approaching fire front. The image in the bottom right is a species of Heterosigma, which we're quite interested in. It's a species that we see down here. It's quite a problem for salmon farming industry. Heterosigma will get into the gills of salmon and ultimately cause mortality. But we also see Heterosigma as north as North Queensland, and so the prawn farming that happens up there, they keep a close eye on Heterosigma. So it's a strain that we have in our collection and that we're looking at initially. So we have a range of different imaging systems. We have a lab-based Zeiss Axio Observer microscope, which is a light microscope capable of imaging in high resolution RGB images, as well as other wavelengths such as ultraviolet and blue violet. We also have access to an imaging flow cytometer, which is a slightly different instrument. It captures images fairly low resolution and in grayscale, but it's highly autonomous. It'll draw a continual sample of fluid through an imaging plane and does a fairly good job of isolating individual cells. But this focus will mostly concentrate on light microscopy. So the early work for this project has really been around mastering our instrumentation and coming to terms with the different variables and factors that are available to us. A couple of those are magnification and contrast. We have a number of different objective lenses on our microscope, and we can image at 200, 400, and 630 times magnification factor. And similarly, we have a range of different contrast settings, so we can capture imagery in bright field, differential interference contrast, and phase contrast. So in each of these cases, you can see different details and different image features coming out. And in terms of building up a machine learning training set, I guess the ideal scenario is that we don't want to have to build up a huge training set with every combination of these factors and other factors. It'd be nice if we could focus on one magnification factor and one contrast setting and be able to train machine learning models that can then make predictions across other magnification levels and contrast settings. So there are a number of other battlegrounds that we have. Motility, cell motility is one of those. So we're working with a living collection here, and some of the species that we're interested in are quite active, specifically the dinoflagellates, which have a flagella, which is effectively a tail that they use to motor around with. At the top left, we've got Alexandrium minitum, which is probably the most active and motile species we've imaged so far. That's a fairly low magnification image, and you can see how active they are. And as you go to higher magnifications, they become more and more difficult to image. We have in the top middle a single Heterosigma carterae cell that we're chasing around with the microscope while doing a video capture. And at the bottom, we've got a couple of different Heterosigma strains at different activity levels. At the bottom left, this is actually really good and interesting for machine learning. We've got quite a few cells that are rotating and turning in the focal plane, which is actually really good because we're getting a really huge number of perspectives on the cells. So when we go and annotate these images, we can actually build up, the machine learning models will be able to build up quite a good three-dimensional representation of the cells. On the right-hand side, we've got focus. So for cells that are not too active, we have been playing with rolling the objective lens through the cells, and you can see a lot of the intracellular detail that we can draw out with this technique. The image at the top right is actually Alexandrium minitum again, which is our active species over on the top left. But in this case, we've added just the lightest touch of lugols, which is a fixing agent. You might use lugols if you're collecting samples in the field and you want to fix them for transport or fix them for long-term storage. One of the issues with applying lugols is it has an effect on the morphology of the cells. So you can see these cells are a little bit almost damaged, especially this cell up at the top here. And the more lugols that you add, the more the cells will, or the morphology will change. So we're still pondering how useful that's going to be for us. So we're now building up a machine learning training set. We have a database of images. The next step is annotation. So we've been using a platform called CVAT, which stands for Computer Vision Annotation Tool. It's a platform that's built by Intel, and it gives us access to a number of interfaces that are useful for annotation. It's actually got some AI tools inbuilt in it, which are machine learning models in their own right. And we've been using a model called Dexter to segment some of our imagery. So Dexter allows you to provide the leftmost, rightmost, topmost, and bottommost points of the target that you're looking to segment. It then goes and queries a pre-trained model that's been generalized to and trained on a very large body of imagery to produce segmentation masks. And that'll produce a bounding polygon for our image, for our algae cell. Then you can change the number of points, and so that's something we've been using to effectively annotate our data sets. So you may have seen this image before. This lays out a number of sort of common computer vision tasks. From the left, you might have a classification problem where you're dealing with a single object in an image. Or you might have semantic segmentation, which is no objects, but each pixel gets a label. Or you may have classification plus localization. So the image has a single object in it and you're localizing that image to an area within the larger image. We're more interested in the two on the right, object detection and instant segmentation, where you're detecting multiple targets or objects in the image, and they can potentially be of the same type. I mean, one of the ultimate aims of this work is to be able to use a microscope platform to image slides that have counting chambers, which have a known sort of volume of sample, and then be able to count all of the cells in that counting chamber and classify all of the cells in that counting chamber. And with that capability, we'd be able to determine what species are available in the environment and their concentration, so whether they're at a concentration level that's going to cause harm or not. So with this in mind, this is the network architecture for a masked RCNN model, which was an early and fairly popular instant segmentation network. And I won't labor too much on this slide other than to point out at the start of this network, there's a CNN block, which contains a number of layers called a feature pyramid network. And the aim of that is that it breaks an input image down and downsamples by a factor of 2 in every layer, and allows you to produce a multi-resolution view of your data with which you can train feature maps on. So this is important to us because ideally we would like to be able to train our models on maybe 1 magnification level, say 630 magnification, but also then be able to make predictions on the other magnification factors. And this is a reasonable structure that may be useful for that. I'll jump right over to the right-hand side and just talk quickly about the outputs of these networks. Typically, you'll get predicted classes for the number of objects that are detected in the input image, and you'll get a bounding box for each of those objects. And finally, you also get a segmentation mask, just like the one we were drawing in the earlier slides. So we anticipate customizing the heads of this architecture such that we can produce a concentration for the entire image for each of the detected classes. So just looking at some very early results, we've now built up an annotated data set, and we're training models on a relatively small data set. We're talking at about 500 segmentation masks for a particular species. In this case, we're looking at Heterosigma again, and we've trained our model on just the 630 magnification factor images. Or segmentation masks, and then we're testing it on the left here on imagery at the same magnification factor and at lower magnification factors, and we're getting fairly good detections across these images, and the confidence strength of those detections is fairly high at 630, still pretty reasonable at 400, although the confidence level is dropping a little bit, and then when we look at testing on magnification at 200 times, we're picking up the larger images, the larger algae cells, but missing some of the smaller algae cells. And the confidence that we're seeing in those detections has degraded somewhat as well. So what's happening here is we're really reaching the limit of the number of layers in the feature pyramid network we were talking about earlier. So we're anticipating customizing that network and adding a few more layers in so that we can train on our high resolution data and then still be able to reach a much lower magnification imagery and make confident predictions. And as a secondary result, we've trained some models and tried to do a sort of cross-domain detection for different contrast settings. So these images here were trained on phase contrast imagery at 200 times magnification, and then tested on bright field imagery and tested on DIC imagery. And we get fairly good detections across the board and fairly high confidences in those detections. So that seems to be working well. And here is a similar result. Models, mask RCNN models that were trained on 400 times a magnification factor and then tested on different contrast settings. And it seemed to work quite nicely between DIC and bright field. We did have some missed examples in DIC to phase contrast. And so that's going to be something for us to follow up on. But in most cases, it looks like we will be able to take this multi-resolution approach and be able to handle different magnification factors and different contrast settings, which is really important for us to be able to build up our machine learning training data set, but then also be able to deploy ultimately on microscopes, compact microscopes that you can take into the field and operate in the field and get detections in effectively near real time. And so that's it for me. Thank you.
Thanks, Chris. If you wouldn't mind stopping sharing, great. And if I could just invite Pete and Nicole to turn their cameras on, or Pete, we'll see how your connection is going. We might have you looking anonymous there. And James as well, sorry. And so thanks very much to all our speakers. Hello. And yeah, if anyone did have any questions, we are hitting up the end of the session we had scheduled today, but I am happy to go just a few minutes over time to see if there were any questions. I wouldn't mind actually just reaching out initially to Nicole. I hope you don't mind me putting you on the spot here. Nicole, she has a background in entomology and spent a considerable amount of time working with the Australian National Insect Collection and is now the Digital Collections Manager for National Research Collections Australia. You're someone who has seen digitisation go all the way through from, taking photographs down microscope cameras to these sorts of applications. Do you have any observations on the most exciting opportunities you see in this area?
Good question to put me on the spot.
Sorry.
No. I would probably definitely say the throughput. you know, the opportunities in the years to come, if we can ramp up on the throughput of, I mean, we have large biological collections here in Australia, and if we can sort of get to those images and data to get it to the researchers, I think that's our biggest legacy that we can leave people with, sort of thing. While, like, I'm actually here in the herbarium collection right now, and while the collection sits in compactuses and then stored away like this, if we don't yet have the digitized data there for people to use, like the postdocs here, it's, yeah, it doesn't have the impact that it can have. So I think that's what a lot of Chris and Abdo's work will show in years to come.
Yeah, realising the impact of these collections. That's fantastic. We didn't have any further questions. James, did you have any questions for our speakers, or? Well,
not from me. I haven't seen anything come through.
Oh, I've got one here.
Oh, hang on, there is one.
Yep. So for a question for Pete, what kind of metadata are you using for curation and digitisation?
What kind of are you referring to the data associated with individual specimens? Is that what you're referring to?
Yeah.
Yeah. I'll say something about it. Nicole will probably be able to sort of talk about this one in more detail, but there's all sorts of information potentially associated with specimens, ranging from obviously. Can you hear me?
Yeah, sorry, I just stopped your video because you were getting very wobbly. Could you perhaps start?
Right, okay. So there's yeah, no worries, that's fine. So there's all sorts of information potentially associated with individual specimens, ranging from obviously the basics like where and when it was collected, in some cases what the plant community was around in that area, the life stage of the specimen. Increasingly, we are developing other data layers that are associated with these specimens as well. As I said, genetic data in many cases, tissue samples. In the case of the fish collection, we're looking at things like, you know, X-ray data and so on. There's all sorts of other data layers as well. So I guess the ultimate goal is to be able to integrate all this data, to be able to sort of simultaneously look at it in the context of a specimen. Nicole, you may wish to add some more to that. There might be other things I hadn't thought about.
I wasn't quite sure what, like, was the question also targeted at how we curate the metadata, as in Darwin core fields, and how it's standardized?
It looks like we do have a sort of a question in that area. I'm not sure if that was the original question or not, to be honest. I guess the first thing is you know, what other sorts of metadata are associated with specimens, and then there was a question about how important are standardized vocabularies to assist in identification. I mean, obviously, it's important. I think, well, I'd say standardized vocabularies are quite important. One of the challenges that we face, I suppose, is that each of the different biological collections in CSIRO have some differences in the vocabularies that they use, I suppose, or how they manage their databases up to this point, because each of the biological collections in CSIRO have developed somewhat independently in different business units or different parts of the organization. Under the sort of National Research Collection, I guess, umbrella, we're bringing all those collections together and trying to unify the way we manage the data, the way we talk about it, the way we sort of record it and store it. So in fact, and I didn't mention this earlier, we're now in the process of actually migrating all of the databases to a single collection management system called Specify, which will mean that we'll have all of those collections sort of unified and accessible within the one platform, which is certainly an important part of standardizing things. I'm not sure if that answered your question or not. Quite possibly not.
Yeah, I think what we see is that standardized vocabulary is essential for this kind of sharing, but then when you try and share across collections with those sort of host species. And,
it can be, and it's challenging in other ways. Yeah, because different collections collect different kinds of things. So the plant collections are primarily pressed dried specimens, but the vertebrate collections, there's skins, there's stuffed specimens, there's large collection of specimens in alcohol, there's the egg collection, there's tissue collections, and then insect collections are yet another different thing altogether. And then of course the algae collection that Chris was talking about is a living collection. So again, they're all very different kinds of materials.
They really are. It's funny. I might actually wind us up with those questions there because we are losing quite a few people. We have run a bit over time. I did love the videos Chris included. Motility is not usually a issue we need to think about in terms of library collections, unless we've got a real problem. Thank you. I'd like to thank all of our speakers so very much today. It was a really interesting session. I was a bit dazzled by some really amazing applications you're putting these collections to, and I really love to see the end result of these projects. I'd also like to thank James, Ingrid, and the rest of our coordinating group who are working with the AI4LAM Australian New Zealand community and our attendees who came along today. If you are interested in more information about AI4LAM, please go to AI4, that's the number LAM.org. That's the way you can join the mailing list. You'll also see we've got a Slack channel and we have an Australian New Zealand chapter channel in that Slack workspace. There will be probably one more session for AI4LAM this year, but it'll probably be a social Christmas get-together kind of session. And we're thinking about projects to showcase in this webinar series next year. So if this excited you, if you've got something you're working on that you'd love to share with this community, please reach out. We're fairly easy to find for that website, and we would love to hear from you. Thank you very much. Thanks for your patience in going over time, and I will see you all next time.
Thanks, everyone.